National Advisory Eye Council, 6700B, Rockledge Drive Conference Center, National Eye Institute, NIH, June 21, 2019. Strabismus, Amblyopia, and Visual Processing, SAVP, Programs, Development and Regeneration, Central Visual Processing, presented by Martha Flanders, Ph.D. Video begins with view of Dr. Anne Schaffner at the council table, followed by slides interspersed with views of Dr. Martha Flanders at the podium in the front of the conference room.
Our next speaker is going to be Martha Flanders. Martha is a program officer at the NEI, and she oversees a portfolio of grants in central visual processing.
First slide shows list of SAVP portfolios separated by program director, boxed in yellow, Development and Regeneration by Tom Greenwell, Ph.D., and Central Visual Processing by Martha Flanders, Ph.D., not boxed, Ocular Motor Systems, Strabismus and Amblyopia, and Sensory Neuro-Ophthalmological Disorders by Humam Araj, Ph.D., and Perception and Psychophysics and Myopia and Refractive Error by Sherry Wiggs, Ph.D.
SAVP. So it's a real pleasure for me, really an honor for me to tell you about this great group of portfolios called Strabismus, Amblyopia, and Visual Processing. So this is really a cluster or a group of portfolios, and I am the leader of this group, and we've decided as a group that we're going to present part of it next October and have an invited guest, and the other part here, the bulk of it is really this central visual processing. The cluster of portfolios is about visual processing and disorders that influence it, like strabismus and myopia. And it's also a group of portfolios that range from the very cell and molecular approaches in the developmental portfolio and to psychophysics. So it's really a broad range, which is partly why we decided to break it up into two. And so what I want to accomplish today is just give you a feel for what goes on in the portfolio, in these two portfolios, developmental and the central visual processing. And what our principal investigators, what drives our PIs, what are they thinking? So what's that portfolio composed of?
New slide showing pie chart of NEI research project numbers within each group of portfolios. SAVP is highlighted with a yellow star and takes up approximately 20% of the pie.
So just to step back and get the big picture, this is drawn from the FY18 awards in research project grants. And you've probably seen this pie chart before. Retinal disease has the biggest number of research grants funded. SAVP is the second biggest, and it actually in this recent count it's shrunk a little bit from previous years, and I think that may be because a lot of these investigators from SAVP are the ones getting the brain funding. Now you know and you hear again and again that brain is a very important part, and the Eye Institute is a very important part of brain, is what I should say. And many of our SAVP retina, but also SAVP PIs are being funded by the Brain Initiative. What we learned last time in January is that many of the brain awards go to NEI PIs. And we also may have mentioned that the working group that's provided the vision for the Brain Initiative, the first one, and then now the update of that vision, were co-led by SAVP PIs. So Bill Newsom for the original vision of brain, and now John Mansell for the update of that vision. So vision in neuroscience has really always led, in my opinion, and you see it very much in brain.
Slide shows pie chart dividing SAVP grants by portfolio. Development and Regeneration, approximately 10%, and Central Visual Processing, approximately 60%, are highlighted.
So what I want to do next is zoom in on the SAVP portfolio and give you yet another pie chart. You can see that the Central Visual Processing portfolio is the largest. And in fact, I've left it empty now in the pie chart because in future slides, I'm going to subdivide it yet again. But this is showing you that the other portfolios, myopia, oculomotor, and so forth, are going to be saved for fall. And today in the yellow boxes, I'll be talking about the development and regeneration portfolio and the central visual processing portfolio.
Slide listing the topics included in the development and regeneration portfolio and the central visual processing portfolio.
So as an overview of the presentation, I'm going to start with development and regeneration, which is actually Tom Greenwald's portfolio, and he's here, I hope, to answer your questions there in the back. So I'll just give you really a snapshot of what's in that portfolio, starting with axon guidance, synaptogenesis, synaptic patterning, so the kinds of synapses that are forming on these postsynaptic neurons. As a basis for that patterning forms the basis of complex visual functions, as I'll try to explain, and as a substrate for plasticity, which I'll talk about a bit. And now when we transition into visual function and like experience-dependent plasticity, we're really transitioning into the central visual processing, my portfolio, when we start to talk less about development and more about function. So within my own portfolio, central visual processing, I'll give several examples. As I said, I want to share with you sort of the motivation and the thinking of the PIs in this portfolio rather than go through all the details of what they do. So I'm going to talk about the thinking in terms of trying to get at the biophysical basis of neuronal computations, and the idea that there's sort of a progressive feature extraction as we go from retina to V1 to higher levels. And then I'll get into some examples of complex visual motor functions from projects in the portfolio and even brain-machine interface. I'll be showing you.
Slide showing 3 cartoons. On the top, showing axon guidance. Left cartoon, uncrossed, shows a neuron from the VT crescent being repelled at the chiasm by Effrin B2 and SHH. Right cartoon, crossed, shows neuron from non-VT retina proceeding through the chiasm to contralateral side. In the chiasm, attractins VEGF and SEMA6D, permissive NR-CAM and plexin A1, and growth-promoting VEGF. Bottom cartoon, synaptogenesis, shows connection between presynaptic bouton and postsynaptic spine connected through major histocompatibility complex class I molecules and PIR-B receptors. Artwork from Trends in Neuroscience.
So starting with axon guidance, this is the part of Tom's development and regeneration portfolio that our own Carol Mason is funded through. And I just have a quick snapshot of some of the factors involved in the guidance of axons as they come out of the retina and try to find their way to the appropriate places in the brain. The kind of retinotopic mapping that's maintained in that process is also covered in this portfolio. For instance, the Effrin gradients in the superior colliculus, that would be in this development and regeneration portfolio. Also, the cartoon on the bottom is showing a picture of synaptogenesis. And I've chosen a cartoon here that shows some of the important players in synaptogenesis. Synaptogenesis, of course, is one neuron contacting another after it's grown out to find the right neuron to contact. So you see on the right, the postsynaptic spine, the dendritic spines are the places where the synapses are forming. You see glial cells bracketing the synaptic cleft there. Glial cells are often important players in getting these in synaptic plasticity and getting the synapses formed and getting them formed right. I also wanted to mention the red squiggly things are molecules of the major histocompatibility complex. And these are players in the story about the PIR-B receptors that has been made so famous by Carla Schatz. And so as you probably know, the PIR-B receptor is important in synaptogenesis or synaptic plasticity, especially in cases where you're trying to reopen the critical period for amblyopia therapy.
Video transitions between slide and view of the council room. Slide shows figures from the article titled LRRTM1 Underlies Synaptic Convergence in Visual Thalamus from the Michael A. Fox Laboratory, published in eLife in 2018. On top, a cartoon showing ganglion cell axons extending from retina to areas of the brain, the SCN, VLGNE, the IGL, the DLGN, and finally the SC. Bottom, 3 electron microscopy images color-coded by neuron, and images of 3D segmentation showing simple, with single retinal terminal per dendrite, and complex, multiple retinal terminals per dendrite, synapses in the DLGN.
Okay, good. This is where I really needed the pointer, because I want to draw your attention to this complex synapse. So now we're on to the topic of synaptic patterning. I mentioned that after you have the axon outgrowth and the mechanisms, all the molecular mechanisms for synaptogenesis. Now here's a recent study from the Fox Laboratory where the investigator is starting to look at whether or not these are just simple one-on-one, one retinal terminal in this case. Here's a simple pattern of synapse between the retinal ganglion cell axon coming in here to the dorsolateral geniculate nucleus. Some synapses are simple, some are much more complex. So here you see color-coded that the different retinal terminals of different types have come on and converged on these postsynaptic neurons. So the hypothesis tested in this recent study from Fox was that, first of all, that what is the function of having these complex synapses? Does it matter to the animal? So what they did in this study was they made a knockout mouse that was lacking a synaptic adhesion molecule, and they found that in these knockout mice, they could indeed wipe out the complex synapses, but not the simple synapses. And these mice did indeed have they had vision, they had visual acuity, they were tested on the Morse water maze, they could see a vertical striped indication where the platform was, so they could perform simple visual tasks with just the simple synapses. But as soon as the task got more complicated, like distinguishing a vertical from a horizontal gradient to tell where the platform was, then these mice that didn't have the complex synapses performed significantly worse. So the message being that maybe there's an association between the complex synapses and complex visual function. So what are the implications of this? Well, one obvious practical implication is if we want to regenerate the axons, maybe we need to worry about getting the right patterns of synaptic connection if we are to restore normal visual function. So that's a practical implication. But why do we have these complex synapses anyway? And here I want to take you to another study, and this time we're in the central visual processing portfolio. This is a recent study from Chen and Anderman in Cell, pretty recent.
Slide showing cartoon or data from manuscript titled A Fine-Scale Functional Logic to Convergence from Retina to Thalamus by Liang et al., Chin-Fei Cheng, and Mark L. Anderman Laboratories. Published in Cell 2018. Left cartoon shows laser pointed at thalamus. Right cartoon shows 3 schematics with different combinations of axis preference, vertical or horizontal, or with on preference, retinal axonal boutons.
And what they're doing in this study is they're looking closely again at these boutons from the axons of the retinal ganglion cells that have come in here to the lateral geniculate of the thalamus. So they're looking closely at those complex synapses, and by wonders of technology, they are actually able to quantify the function of each individual bouton. So the mouse here in the study is being shown patterns of visual stimulus that are either coming on or going off or on off, and this is to characterize whether those incoming axons are from cells that have this on or off characterization from their properties in the retina. So there's different, different properties for different neurons. Other neurons coming from the retina might have this property of preferring a certain direction, horizontal, vertical, or oblique. The direction of motion in the retina would be a functional a characteristic that the retina is carrying to the LGN. And this cartoon shows various patterns of convergence. We know we had complex synapses, but is there a reason for that? Well, you see various possibilities here. You see possibilities where, for instance, all the vertical direction, all the neurons that prefer vertical or horizontal direction of motion are coming together and they're combining but they could have different on-off preferences. All right, so there's this pooling of information, and they quantified all the different kinds of patterns. Here's another example where it could be any direction of motion preference coming in, but it's only the on axons that from the ganglion cells that prefer on. So you have really a structure here that picks out certain functions and makes it invariant to other things. So it's feature invariance. You have the kind of pooling of information that will let a postsynaptic neuron in the LGN be sensitive to horizontal motions, no matter whether they're on, whether the light is coming on or off. So this kind of invariance is sort of a functional explanation for this what seems like a complex hardwired circuitry that's come from development. Okay, was that clear? So you, however, if you think a little bit more deeply about this, and these authors, so we need to go back. These authors did mention, thought a little bit more about it and mentioned in the discussion of the paper. So you have some functional logic, and some functional logic is what they called it, reasons for these patterns, for information processing. But it also, they mentioned, would provide the substrate for plasticity. So now imagine, this is what the brain does. This is what brain circuits do, is there's an opportunity for changes. When we learn things, when a circuitry is changing for various reasons, you want to build plasticity into the system. And they mentioned that having hardwired, complex, convergent patterns would actually give the opportunity for plasticity. Imagine that you could enhance some synapses but not others. That would actually change the functional characteristics of the cell.
Slide showing data from manuscript titled Differential Tuning of Excitation and Inhibition Shapes Direction Selectivity in Ferret Visual Cortex by Daniel E. Wilson, Benjamin Scholl, and David Fitzpatrick, published in Nature 2018. Top, 2-photon calcium imaging, approximately 300 by 300 microns, with spine labeled with red and blue dots. Bottom, histogram labeled soma with x-axis of angle, right, y-axis of delta F over F, peak to 0.5 at 270 degrees, in the upper left corner of graph, image of cells.
And this same type of thing, imaging the functional activity in dendritic spines, is also done in visual cortex. So I've now taken you from the lateral geniculate to the primary visual cortex, and through this amazing technology, and this one is from the Fitzpatrick lab, now you see a case where with 2-photon calcium imaging, the calcium imaging shows the activity in a neuron usually, and this chart is from the cell body, so you can see the activity of a neuron in response to a visual stimulus being given to the mouse, the activity of a neuron in the soma, but you can also quantify the activity in individual dendritic spines, which is what's shown up here. So this is an example where the ferret this time is being shown motion in different directions, and you see the angle, the direction of motion here. And now they've quantified activity in the cell body of a pyramidal neuron in mouse visual cortex. So head-fixed mouse, but the visual stimulus is moving in various directions here. And we can say that this pyramidal neuron that we're looking at is preferring the 270-degree direction. So this is the directional preference of this neuron. But now look at the dendritic spines here, and you see that color-coded in blue, the cell body likes 270 degrees, and some of the dendritic spines are actually most active for that direction, but the ones coded in red are active for the opposite direction, in fact. So there's this, even we can quantify, and I actually think this is most exciting just to be able to quantify this in a living visual cortex while the alert animal is watching stimulus on the screen. So now this provides the opportunity. I think that the biggest thrust of this Nature paper was that we can do this in mouse primary visual cortex. And now this gives the opportunity to pursue all sorts of questions of plasticity in the circuit of the primary visual cortex.
Complex diagram from manuscript entitled Amblyopia, New Molecular, Pharmacological, and Environmental Approaches by M.P. Stryker and S. Lowell, published in Visual Neuroscience, 2018. Graphic shows how pyramidal cells are affected by dark exposure via numerous pathways, including the thalamus.
And I'm not going to take you all through this, don't worry, but it's from a review article from Michael Stryker, and I actually found it very helpful in trying to understand what was going on in my portfolio, because there's a lot of activity in this area, and people are looking at the different things that are being modulated. I put in the yellow box. They are really the target of all this modulation. So you're seeing dendritic spines. You're seeing synapses that want to be unsilenced or subject to spine density, so the new spines grow, subject to some form of plasticity. So you might want to have some circuit that helps with synaptic plasticity in various ways, either by unsilencing synapses, promoting synaptogenesis, or even just heightening the activity or the responsivity of an entire neuron so that that pyramidal cell can form this pyramidal cell could possibly enhance its synaptic connections to other pyramidal cells in the circuit as a substrate for memory formation. So plasticity in this system is key. And just listed here are some of the ways that investigators in the portfolio are looking at acetylcholine receptors, even transplantation of embryonic inhibitory neurons. But the big picture really is that it's this local circuit that's promoting, that's feeding into the pyramidal neurons, the key players, and really modulating the excitation inhibition of this system and giving rise to plasticity. So the punchline here is that V1 experience-dependent plasticity is a local circuit phenomenon. There's lots of players and there's lots of people, lots of PIs and projects in this portfolio that are looking at plasticity in very detailed ways.
Pie chart showing breakdown of central visual processing portfolio into topics, from largest to smallest, cortical representations and processing mechanisms, structure function of the cortex, higher cortical functions, structure function of the thalamus, higher control of saccade, V1 plasticity, and other. V1 plasticity and structure function of the thalamus are highlighted, then later the remaining sections, except other, are highlighted, and then video returns to view of Dr. Flanders.
However, this V1 plasticity is only a relatively small part of the portfolio, so I want to move on now and talk quickly about some of the other parts of the portfolio. So V1 plasticity, now I've made a pie chart to help me understand my own portfolio. I've broken it down and I went through all the grants and broke it down into various areas. I've already given examples of V1 plasticity and the structure and function of the thalamus. This is going to include also the pulvinar, important area of the thalamus, and we have studies of the portfolio that are looking at connections between the thalamus and the cortex and back. So the thalamocortical circuit is represented here in this portfolio. But the bulk of the portfolio is really some of these other areas that are very basic fundamental research that try to get at visual processing, the anatomy of the system, structure, function of the cortex, visual cortical representations, processing mechanisms, higher visual cortical functions, just a really wealth of Hubel and Wiesel type work that's going on in this portfolio, and very, very brain initiative related as well. So this is, as I said, this is basic fundamental research. It underlies disorders like amblyopia, various agnosias would be served by understanding this vast literature. But more recently, what's really come across is that in recent efforts for visual cortical prosthetics, so Brain is funding 2 projects on visual cortical prosthetics, where there are electrodes being inserted into the brain of blind subjects, and they're trying to figure out the correct pattern of stimulation to use there to restore some kind of vision. So at this point, all of a sudden we start to appreciate it's a good thing we know something about visual cortical processing, because there's so much that needs to be done to fine-tune the right patterns to restore vision. For example, the issues that have come up right away is you can't always get the right stimulation site. The electrodes sometimes have to be implanted not in the foveal region of V1. It's going to be someplace else. So it's a good thing we know the anatomy. So the stimulation timing is critically important, because there's plasticity in this tissue. Also, there's adaptation of this neuron. So if you're programming in, you have a camera on the head, mounted on the head, and you're trying to use that image to figure out what pattern of stimulation to give, it's a good thing we know something about what kind of stimulation that circuit naturally gets from the thalamus. And then the one thing that really has come across is eye movements. All of a sudden, the camera's on the head, the visual cortex is naturally getting signals that where the retina is moving all the time, the camera is not moving. What do you do about eye movements? So it just makes me glad that we've done so much basic research on eye movements, because here's a problem that really needs to be tackled.
Slide with text surrounding an artistic representation of two neurons interacting. Artwork from online-learning.harvard.edu.
So I thought that there's so much in this portfolio that I thought maybe I'd back up a little bit and just sort of start it out by giving you, as I said, I want to give you an understanding of what people in this portfolio are thinking. So I thought I'd sort of break it up into approaches that follow sort of are looking for explanations in terms of the biophysical basis of neuronal computation. The portfolio is all about visual processing, visual computations. The scientists in this portfolio aren't happy unless they can envision a mechanism at the cellular level for the visual perceptual phenomena that we're trying to study, basically. To put it simply, everyone seems to have in mind the cell morphology of the neurons, the membrane properties of the neurons, the synaptic connections in the local circuit and beyond. Those are the basic things that we want to have in mind when we're measuring. In the lab, we're able to measure things like orientation, orientation, the response of a neuron to the orientation of the stimulus, to the direction of motion of the stimulus, to the contrast, to the spatial frequency, to the binocularity. Those are things we can measure the response of a neuron. We can give stimuli in certain, very carefully controlled directions or of different spatial frequencies. But we'd like to understand and relate it back to the cell morphology, which actually determines receptive field size. The membrane properties determine the dynamics and how quickly adapting a neuron. And so we talk in the PIs in this field talk about the neurons tuning. What is the neuron like? What is the neurons tuning, or what are its preferences? So this is one sort of thing that's in most of the PI's heart, is to trying to understand it in terms of the biophysical mechanism. And these two conceptual approaches are not mutually exclusive. I think everyone subscribes to the theory that's in every textbook, that when we're thinking about visual processing, we're thinking about sort of a hierarchy of features presented on the retina, which follow through to relatively simple features presented in primary visual cortex. And then when you go on through the visual pathway, when you get, for example, into temporal cortex, you have more complicated figures like shapes and faces. So this is hierarchy built in. And I'm willing to call this a theory, because I think everyone would sort of, this is always in the framework of our thinking.
Slide with black and white Ansel Adams photo, The Tetons and Snake River, 1942, at the center. In the background, dark peaks capped with bright snow and luminous clouds above. In the foreground, dark hills to left and flatter ground covered in trees to the right are bisected by bright curving river. To the right of the image, graph with x-axis object luminance and y-axis neural response. In solid line, -45 degrees from top left to bottom right of graph, the off pathway. In dotted line, increasing quickly from bottom left and tapering off at upper right, the on pathway. Reference Principles Underlying Sensory Map Topography in Primary Visual Cortex by J. Kremko, J. Jin, Y. Wang, and J. M. Alonso, published in Nature 2016.
As a couple of quick examples of these two ways of thinking, conceptual approaches, I thought I'd use some work from our own council member, Jose Manuel Alonso. And if you don't understand this, he'll explain it at lunch. And who's actually, in a beautiful way, used the interplay between characteristics like on or off neurons. Those features of the light comes on or light comes off. So features like on, off, features like spatial frequency, contrast sensitivity, and you see here in the diagram that there are sort of complex interplays between neurons that are carrying that type of information. Even in primary visual cortex, Jose Manuel and his colleagues have in the Nature paper, which I liked very much, they discovered that there were more off neurons in primary visual cortex and coined the phrase dark dominance, which does indeed sound like Star Wars to me. But more than that, they've used it to explain the mapping of features in primary visual cortex. But also, some of these interplays suggest gestalts, like there should be sensitivity to emotion and depth. And the gestalt I got from reading some of these papers was that maybe this explains why some art is more beautiful to us than others, or why some art is beautiful. I wonder how many of us have ever thought about whether or not we're seeing what's actually there, or if we're seeing what our brains are telling us we see. So what followed for me from reading the papers was that objects like large white objects, like here the river or the clouds, look larger and fluffier than they would. He's nodding, so maybe I got this right. And there's also you also have a better ability to see fine details in the dark areas. So I think, maybe Ansel Adams was onto something here.
Center of slide, a painting of a scene with a plaza, buildings on right and left, cathedral in the background, and bell tower background right. Clusters of people are shown in the square. Over the image, 13 fixation points are marked, with lines showing the tracking between fixation points. A zoomed-in view of fixation point 12 shows a red line shifting between different figures in a cluster of people. To the right of the painting, a graph showing eye of movement as a function of time. Fixation points, marked in magenta highlight, show clear sudden changes in direction of eye movement. Reference Control and Functions of Fixation Eye Movements by Michelle Rucchi and Martina Paletti, published in Annual Review of Visual Science in 2015.
Another thing I wanted to briefly mention was I had mentioned the importance of eye movements, and I put this in the category of biophysical explanations. It's been, especially by Rucchi and colleagues, it's been appreciated, again, maybe this is a rebirth of this activity, but as we scan a scene, as you see here, the yellow lines are saccades to different fixation points. Now, ideally, fixation points should be where your eye is still. That's why they call it fixation. But if you look closely at a fixation point, like the F12 fixation point there, and you can see here a close-up of over time, the movement of the eye, and these periods in between the saccades are supposed to be steady fixations, right? And you can see here in the red what's actually happening during what we thought would be a steady fixation is it's as if the retina is rubbing itself across the visual image, much as we rub our fingers across textures that we like to feel. So it's been appreciated more and more, and I think I see this on every study section now, is what about eye movements? Because if the eye is moving, and we've gone to such trouble to quantify the spatial frequency tuning, the contrast sensitivity of a neuron, that changes if the eye is moving. So the image on the retina that's being conveyed to the visual cortex is different in terms of its spatial temporal frequency because of this retinal blur. So people are starting to appreciate that. And again, so it's relevant to V1 neurophysiology. People are starting to account, worry about eye movements and try to take it into account, but also for visual cortical prosthesis, as I mentioned. If the eye is really moving all the time, and if there's purpose to those small eye movements, then we should know that in a visual cortical prosthetic interface.
Slide showing data and cartoons for feature extraction conceptualizations. Center, human fMRI map showing face and non-face patches in red and blue, respectively. Reference, extensive childhood experience with Pokemon suggests eccentricity drives organization of visual cortex by Jesse Gomez, Michael Marnett, and Kalanick Grospector, published in Nature Human Behavior 2019. On right, top schematic of monkey head with arrow pointing from upper back of head down towards front of the head, labeled View and Variance. Below, fMRI scan of monkey brain, highlighting regions in red and yellow. Reference, 2 areas for familiar face recognition by Sophia Landy and Winrich Freiwald, published in Science 2017. Flanders highlights right-hand section with her pointer.
This one can't be skipped because it's the example of feature extraction, this idea I mentioned that everyone subscribes to. And what's shown here is in the non-human primate brain is that there have been face patches discovered that have been well characterized and form a face patch network. And as you go from the occipital representations down here to the temporal lobe, you basically get from a picture-based representation to something that's view invariant. So if you want to recognize a face, it's fine if it's facing forward, but you'd also like to represent it from different views. So somehow the processing in this stream has gotten you to view invariance. And this paper also discovered a new area in the temporal pole from Freiwald that was just active for familiar faces. So it's a very, very beautiful extreme things that this network is doing, which I think Anita is going to talk more about some of these elegant transformations going on in this.
Feed-forward convolutional neural network model. Complex slide with images of scenes, images showing paths around an object in a scene, and graphs. Reference, computational mechanisms underlying cortical representations to affordance properties in visual scenes. By M.F. Bonner and R.A. Epstein, published in PLOS Computational Biology in 2018.
So I wanted to show some examples from what I call computational fMRI. I see I'm out of time, so I won't have time to go through this. But as part of the portfolio, there are some very elegant, this is an example from Epstein, and it was published in PLOS Computational Biology, which is one of the best journals for that type of thing. And it's an fMRI study where they had a gorgeous experimental design where people were looking at different scenes in the magnet, and they were able to determine through use of a convolutional neural network model in a computationally intensive way, they were able to show that this area, the occipital place area, was sensitive to the path, those are the red lines, that the subject intends to take through the scene as they're looking at the scene.
At top of slide, photo of a man seated at a table looking at a computer monitor, with a gray box and wire extending out of the top of his head. At bottom of slide, Utah arrays. Reference, Aflalo, Guam, Cadlick, Rosario, Parachian, and Anderson, Caltech 2019, unpublished.
Well, I'm sorry I've run out of time. I'm going to show you the last slide very quickly. Very recent work from Richard Anderson's lab at Caltech. This is a photograph of the most recent subject in his the BMI studies with permission. You see the subject here who is paraplegic, completely paralyzed, and you can see there's a connector on his head, and he's looking at a video screen. And what's being recorded in the brain is Utah arrays have been implanted in both the posterior parietal cortex and the motor cortex. So those grids are electrophysiological recordings from those two areas from many, many neurons. You see the waveforms are identifying that we really got a lot of different neurons, and we're getting stable recordings from these neurons. In this most recent subject that has been implanted, they have told us that he required very little training. As soon as he was connected to the computer interface, after the implants had had healed and so forth, he was able to move the cursor on the screen with his own thoughts. And this, I think, is an advance. You're probably aware of the BMI technology, but this one has both posterior parietal activity. Posterior parietal cortex is a planning area, a visual planning area for motion. So the subject can think about where to move, and motor cortex activity, again, thinking. But the interplay between the information in these two areas is expected to give more sophisticated control. So, with a little bit of practice and better training of the algorithm, I think this guy is going to be playing video games. And, that's, according to my son, that's what every young man lives for, is to be playing real video games with their friends. So,
Return to overview slide.
I went very quickly through some of these aspects of the two portfolios, just trying to give you a flavor of what's in them. I left a little bit of a gap because our invited speaker, Anitha, is going to tell you more. And for the interest of time, we just move to Anitha and ask questions at the end, or do you want questions now?
Dr. Schaffner.
Yeah, let's entertain questions. The one slide you skipped over that I thought was interesting was you have to tell us how playing Pokemon changes your brain.
Oh, that's the one I left out. So this is the back. It's back. Okay.
View of Dr. Flanders, then returning to slide with human fMRI data.
So I wanted to try to give a good balance of the portfolio. So I want to be sure to say that we have both non-human primate work and we have fMRI studies. And in terms of the face patches, we've had both human fMRI and non-human primate studies from the Livingston lab. The human fMRI is from the Grill Spectre lab. And in both cases, these studies are interested in the development with young people. How we talked about plasticity, how do these things change over time? So in the adult, there's these very well-defined face patches in the network. How do we get there? How does this develop in the young? So the Pokemon paper in Nature Human Behavior is showing you that in the adult temporal lobe, there are very well-defined face and non-face areas. So some areas you see fMRI activity when the person is looking at a non-face object. In some cases, there's very definite face objects. And this pattern is really quite repeatable across subjects. Though in their design, they compared the face activity in subjects who had and had not had Pokemon playing experience when they were younger. And they were actually able to significantly show that those who had played Pokemon had better face representations for looking at Pokemon figures. So that speaks to development. And I left it out because it's very nice, but it's sort of not unexpected with what we know from development. Yes.
View of room, then Dr. Alonso, sitting between Dr. Guiazda and Dr. Stepp, then returning to Dr. Flanders.
Thank you so much for the nice summary of the NEI portfolio on the visual brain. So I was surprised to hear that the section of the visual processing was shrinking. And I wonder if there is a relationship with the moment where the mouse became popular as an animal model in visual processing. And I'm curious to know what are the relative percentages of mouse versus carnivores and non-human primates now versus, let's say, 5 years ago, 10 years ago.
My impression is that that's held pretty steady. Michael had the portfolio before me, so I'm looking at him.
Dr. Michael Steinmetz, sitting next to Dr. Schaffner.
Yeah, so there's been obviously a great increase in the mouse studies. When I first started, there were almost none. In fact, when I was an SRO in the study section, and an application came in that involved mice, the answer would be, why mice? They can't see anyway. Why are we studying them? So there was almost no representation. But this portfolio has one of the highest representations of non-human primate studies at all of NIH. So it has a really intense number of people doing non-human primate. But obviously the number of mouse studies are growing as the basic research is being done to understand what the pathways are and what the representations are. So there is an increase, a definite increase. That's every year getting bigger.
Dr. Flanders.
So my perspective was only over 3 years, and all I was trying to say is that even in that 3 years, I think I've seen more go to brain. We haven't done an analysis of that, but that's why I mentioned it has shrunk slightly in the last 5 years.
View of the room, then Dr. Van Gelder, then returning to Dr. Flanders.
Martha, it's a very impressive portfolio, and the basic science advances are tremendous, but I didn't see a lot of disease-specific research here, and in particular, do you feel that the amblyopia research is keeping pace with some of these basic findings? Is it a competitive pool of grants of the same quality that you're discussing here? And similarly, I didn't see much about cortical remapping and blindness, which is a big issue if you're going to do these prosthetics and someone is blind from retinal disease and their cortex gets remapped, then are you really stimulating a map that's going to be useful and accurate? Are those active areas of the portfolio?
Well, that's a great segue to the upcoming council presentations in October are going to be the parts of the SAVP group that deal with strabismus and amblyopia specifically, and also myopia, and some of the perception and psychophysics, maybe looking at sort of rearranging mapping. We also have the low vision portfolio, so from the issues, some of those issues. So what I presented today was really mostly the fundamental research. And they'll tell you in October that those areas are going strong too.